Okay, I think we're going to get started. We got a good group in here already. Some more can trickle in. So welcome to the QBI Coronavirus Research Group retreat, which is really tailored towards younger scientists, in particular graduate students, and the graduate programs have been made aware of this retreat, and we're in particularly targeting some of the new students that have come in. Obviously, we're in the middle of a pandemic. It's hard for the new students to get integrated into what's going on, and this is a way for them to figure out at least what some of the research is ongoing with respect to COVID-19. And just a brief background about the Qcrg. It started with about 20 scientists, and then it expanded to over 40, pictured here. And this was formed in March, just when the pandemic was reaching California. And these groups have come together, I'd argue, in a very exciting and collaborative and unprecedented way, bringing their different skill sets to the table to try to come up with solutions here with respect to COVID-19. And this group got so large, we broke it into a number of different subgroups, which you're going to be hearing about here this first hour on Zoom, and then after later on in more details from trainees in the breakout rooms in Discord. So in this first hour, each group is going to give a rough overview, a 5-minute overview of some of the efforts that are ongoing in that particular area. And here's a quick summary of the speakers for this first hour. And at the end, Gina is going to just give a quick intro. I think most people have been exposed to this new program, Discord, which I hadn't been made aware of until recently. It looks pretty cool and interactive. But Gina, at the very end, just stick around. She'll tell you just a little bit about Discord if you haven't been familiarized with it before. So I'm going to talk just very briefly about the Qcrg Systems Biology and Bioinformatics subgroup. I lead this with Pedro Beltreo, who unfortunately couldn't be here. And a couple of papers that have been spearheaded from this particular subgroup. One is we've looked at the SARS-CoV-2 protein-protein interaction map, and in collaboration with the drug discovery subgroup led by Kevin O'Brien, we've identified potential drug targets that we looked at in more detail. And then on the right there, we've globally looked at phosphorylation analysis in infected cells and identified kinases, which are misregulated. And again, with the drug discovery group, identified kinase inhibitors, which have antiviral effects. And these works are going to be discussed in more detail in the breakout room, and that discussion is going to be led by Danielle and Mehdi. And I just wanted to briefly here in the next couple of minutes just tell you about some other projects that have been ongoing in this particular subgroup, obviously in collaboration with many other scientists in Qcrg and beyond. And this is where we're trying to build pan-coronavirus protein-protein interactions. So we looked at SARS-CoV-2 initially. Again, you'll hear more about that in the subgroup breakout. But more recently, we've looked at SARS-CoV-1 and MERS and subjected all of these genes with these other viruses to a similar pipeline that we had used for SARS-CoV-2, where we're doing affinity tag purification mass spec-based analysis to generate maps in these different coronaviruses. We've actually did localization experiments as well, looking at the individual proteins, both the tag proteins, and we've also created endogenous antibodies against many of the SARS-CoV-2 proteins as well. Merged this together, and this is led to functional analysis, structural analysis, and real-world clinical data analysis as well. So here's the protein-protein interaction map that Mehdi and Danielle are going to be talking about for SARS-CoV-2, led by Dave Gordon. But then, as I said, more recently, we have a SARS-CoV-1 map and a MERS map as well. And there's a wealth of biology in here, and there's a lot of bioinformatics that has gone into individually looking at these data and then merging them together. One quick thing you can do is cluster these protein-protein interactions, and you can see that the three biggest groups are interactions that are shared here with all three viruses. There's a subset of just SARS-1 and SARS-2, and then there's a group just with MERS. And what you can do with this is some differential interaction analysis that we did in collaboration with Triadica in San Diego, and you can represent these three different categories in one particular map, where the edges here correspond to the differential interactions. So if the edges are black, it means it's in all three viruses. If it's blue, it's MERS specific. If it's red, it's SARS-1, SARS-2 specific. So for example, here you can look at NSP1. We see with DNA polymerase, it's only SARS-1 and SARS-2, but NSP1 only in MERS interacts with mTOR and STAT. Compare that to, say, chitin kinase 2 here with N. These are black edges, so we see them across all 3 viruses. And this is interesting because Mehdi and Danielle are going to speak about how this kinase is most upregulated in infected cells. Then just to give you a sense of some genetic analysis as well, Martin is going to go through this in his subgroup, some of the genetic stuff he's been doing, but just very briefly, what we did was RNAi knockdowns of all the interactions we found with SARS-CoV-2 and deletion with CRISPR and CACO2 cells. In both cases, look to see what effects they have on infection. We got a number of hits here in both of these cell systems, which are represented here. There's some cell-specific interactions as well as some commonalities. And then this is just a summary of the interactions, the functional connections. The blue nodes here correspond to host factors when deleted, have a big effect on infection. And then I'm showing you here if they're interacting with specific viral proteins with SARS-CoV-2 in red, SARS-CoV-1 in orange, or MERS in yellow. And this has led to a lot of more hypothesis-driven research, more detailed mechanistic biochemical structural studies from other subgroups with respect to the Qcrg. And Clem and Oren are going to talk about ORF9b and TOM70 in more detail, for example. So I'm just giving you a summary here of the large-scale data sets that are being generated that are being used to fuel more targeted studies that you're going to hear about over the next hour. And just the last point I want to make here is that these are very highly collaborative projects. These are the number of authors here associated with this next paper. And for me, the big thing, very exciting thing about QBI and Qcrg is how we're bringing people together, as I said, in a very unprecedented way, very collaborative way. And this is the kind of science I want to be involved in the future, bringing people together with different disciplines, with scientists around the world. And it's just you can showcase how fast you can move scientifically when people can work together on so many different levels. So this is a paradigm that I hope stays in place as we tackle other disease areas in the future. And just the last thing I'd say is just to thank the QBI media team, Jacqueline, Gina, Alexa, and Carolina. They adapted very quickly to the pandemic, and they've learned this new Discord program, which I think is very exciting and very interactive, and you can hear about. Gina's going to give a quick summary of this at the end of this particular hour. So with that, I will hand this over to, I think it's Clem and Oren for their brief overview of
Thanks, Nevin.
Structural Biology Subconsortium.
So I'm Oren Rosenberg, and my partner in crime is Clem Verba. And we got wind of these beautiful maps that Nevin was producing, as well as many other exciting structural biology targets that were being explored around the campus. And we said, we can't do this by ourselves. We've got to get together as a group with all of the superpower structural biology that we have here at UCSF and work collaboratively. And that's what we've done. And so we're really excited to share with you today some of the results of our collaborations over the last 4 months. And I want to just emphasize, and Clem is going to take over in a minute, but I really want to emphasize how all of this work was done collaboratively. This is not work from somebody's lab. This is work from a new group that was formed just for this purpose, to fight the coronavirus outbreak using our structural biology skills. And I'm going to turn it over to Clem.
Cool. Thanks, Warren, for the start. So yeah, so our subgroup as part of the Qcrg, we're interested in bringing molecular details to this protein-protein interaction map networks, which Nevin has talked about. And so now we have a parts list of who interacts with whom. How does this virus come in and hijack the human cells to exploit it for its own purposes? But as structural biologists, what we're interested in is how does it do it? And knowing that will allow us to inhibit it and target it. And although it's a very, it's obviously a super important, pandemic, and there's been a huge, great strides of structural biology in terms of understanding how these proteins work. Many of them are still structural and characterized. Furthermore, all of these host viral interactions structurally basically are completely unknown. We have no idea how these proteins come together to work together. And therefore, we can't really, it's very difficult for us as scientists to engineer ways to break those interactions apart. And as Oren was talking about, we realized very early on that it's not something, going through this 50, 60 proteins is not something we can do one lab or two labs or three labs can do at UCSF. So we actually made a call to all the structural biologists at UCSF, and to our, to great pleasure was that it was over 60 people who actually came forward and said, hey, we want to work on this. We want to work with you together and use our, apply our skills to fight this pandemic. And these are all the trainees and scientists and postdocs and grad students in these labs, coming from 18 of these labs. And this is, to put some sort of personal faces to the meetings. This is a little bit outdated at this point. This consortium is a living thing. Some people join, some people leave. But overall, it's, it's still definitely going strong. And so the next question was, well, now that we have this large group of people who all want to work together, how do we organize this effectively to go for our goals, to understand this virus? And so we broke it up into groups. So there's a leadership group, which is sort of more focused on more strategic, figuring out how to strategically, what to go after as a consortium. But then we have the expression groups in bacterial and mammalian cells, protein purification group, crystallography, cryo-EM group, and data processing and model building subgroups. And each of these groups consists of about, 10 to 20 grad students or postdoc or scientists, and there is a faculty mentor, and there's also a number of team leads who sort of take on more responsibility within each group to really to oversee everyday sort of and help with everyday management. And so what's really amazing is that in this 5 months since basically, where is that? In 5 months since that, since the inception, since basically going from zero, we already got many actually structures of SARS-CoV-2 spike protein with a number of engineered antibodies with collaboration with Zimanglik at Walter Lab, and also similarly engineered a different type of engineered antibodies as a collaboration with the Wells Lab shown here. And on top of that, we also already gotten, as Nevin brought up, we already got one structure of host viral protein, which is completely de novo structure for both of these. We got already another viral structure. And here we're already at sort of a early blobology stages of getting a complex, another host viral complex. And so, but I've been specifically, I left many details out. If you're interested, please join the structural biology and modeling. Actually, we're sort of doing it together as a breakout room where actually the people who did the work, the trainees in the consortium, will actually talk about all these different projects so you can learn more about it. Thank you.
All right, great. Thank you, Clem. Fantastic. So we'll transition to Martin Kampman, who will describe some ongoing work in the genetic front.
Hi, everybody. I think the genetic subgroup is much smaller and newer addition to the Qcrg than the structural and proteomics groups, but we hope that genetics approaches can really provide a nice complementary view of SARS-CoV-2 and its interaction with the host cell. Basically, the genetics group has two aspects, discovery and mechanism. On the discovery side, we are conducting de novo genetic screens to identify new functionally important interaction partners of the virus in the mammalian cells. So what are the host factors it depends on? And we're very excited to integrate this kind of genetic interaction data with the proteomics interaction data. And again, we think that some of those might be potential therapeutic targets. However, in a kind of reverse genetic approach, we are also excited to work with all of the other subgroups in order, for example, to test some mechanistic hypotheses in cells using genetic tools. When the pandemic hit us, a team of people in our lab was really excited to say, hey, our genetic tools would be a great way to address these questions. And even though all of them have their other projects often related to neurodegenerative diseases or neurodevelopmental disease, they teamed up and worked together. We work in a collaboration with a lot of other subgroups and people listed here. I'm just going to highlight one project that we are very excited about, that is a CRISPR-based perturbation screen to understand host factors controlling binding of the virus to the human cell surface, which we think is a potential therapeutic target. our genetic screens are based on CRISPR technology, either CRISPR knockout or a suite of tools, CRISPR-I and CRISPR-A, that was developed here at UCSF. I was involved as a postdoc in the Weizmann lab, and together CRISPR-I and A allow us to up and down regulate endogenous human genes over many orders of magnitude. And we can do this in a genome-wide manner in a pooled screening format. And what we started with was a screen to ask, what determines binding of spike protein or the spike protein receptor binding domain to human cells? ACE2 is a known receptor, but are there some co-receptor, or what are pathways that control ACE2 cell surface levels themselves? We transduced a Calu3 CRISPR-I line received from the Weizmann lab, and then in collaboration with Jim Wells' lab, who had expressed constructs of the spike protein that we could detect fluorescently, we would then sort cells that express different guide RNAs into high and low binding of spike protein, and already identified a lot of hits, both at lower and increased levels. ACE2 itself was a top hit, so that's a nice positive control, but we found many other genes either in a positive direction, enhancing viral binding or decreasing it, and we are currently mechanistically validating that. But I refer you to coming to our breakout room, and you can have much more in-depth discussions with the people who actually did the work. In particular, Raylin, Avi, and Gokul will be in the breakout room. And any other subgroups, if you're interested, please reach out, and we'll always be excited to chat about potential collaborations.
Thanks, Martin. Great job. So next up, drug discovery, which has been led by Kayvon Shokat and Brian Schreikett, and Kayvon will give a quick overview of that particular subgroup. Oh, you're muted, Kayvon.
Oh,
Yep.
Sorry. You see my screen now?
Yep.
Great. All right. Thank you. It's great to represent the drug discovery group that Brian and I run. And the first activity we participated in back in March was the population of the protein-protein interaction map that Nevin already spoke about, with small molecules that target the host factors that were identified from the mass spec. And you see there are just a host of small molecules scattered throughout the map. And with our collaborators in New York and in Paris, we've been validating whether these have an effect on viral replication, and many of them do. And very excitingly, several drugs that were approved or in clinical trials have now progressed into early stage, or about to progress, we hope, into phase 1 trials in COVID patients. So I think something in March we only dreamed would happen really has come to light. So we're very excited about that. And in the poster session, Tia Tamino will tell you about one project of drug repurposing. That is where we've identified non-structural protein 6 interacts with sigma R1, which Nevin also mentioned. And Tia and Brian's group has found many sigma R1 ligands, and we've been screening those for their replication activity and trying to link those molecules' activity to the dependency in the virus. And then Alyssa Fink will tell you about something that we've started more recently, which is actual computational drug discovery, covalent docking to try to hit the cysteine active site in the major protease. So we're not just targeting host factors, but we're targeting the viral factors as well. What I wanted to do was tell you about how you could use the map, and this is really for the trainees. This data is so rich, allows you to go after almost, I think, any phenotype the virus might control. One of the things we've been interested in, my lab, postdoc Xiang Zhang, is how the virus evades the immune system. We've known that HIV can control so Xiang wondered if SARS-CoV-2 might do the same. And we did this in collaboration with Nevin's lab, and also are currently doing assays right now with Adolfo's lab at Mount Sinai. So this is a story about NSP14 and its host factor, IMPDH2, or inosine monophosphate dehydrogenase 2. So the way some viruses avoid the immune system detecting them is by downregulating class 1 MHC. This is a very smart strategy for the virus because, of course, when the virus goes in, it makes all these viral proteins. These are completely non-self, very foreign. If they were presented by class 1 MHC, they would be seen as foreign, and T cells, CD8 and CD4 cells would be able to kill virally infected cells. Xiang asked, are any of the SARS-CoV-2 ORFs responsible or capable of downregulating class 1 MHC? And he expressed all of the constructs from Dave Gordon in HEK 293 T cells. And the one I'll focus on, you see a subset of them do downregulate, notably NSP1 and NSP14. And so then he asked, now that we know a viral factor that controls class 1 downregulation, how can we intercept that? So we go look back at the protein-protein map. We see NSP14 interacts with IMPDH2. Incidentally, when Adolfo's lab tested all the IMPDH2 inhibitors in viral replication assays, they were completely inactive. So we sort of missed on the first phenotype that we had used to interrogate that interaction. But now, when he uses the IMPDH2 inhibitor to see if it can influence NSP14's downregulation of class 1 MHC, you see on the right, NSP14 expression downregulates class 1 and beta-2 microglobulin, its co-receptor, its associated protein. NSP14 is there. Now we put in mycophenolic acid, we rescue class 1 MHC on the surface and in the Western blot, and we reciprocally destabilize the viral factor. So this is one example of how you can go through the map and look for other phenotypes besides replication. I think it's great. Hopefully you can come to the breakout for the drug discovery and hear the two poster talks and ask us anything else. And please reach out to Brian or I if you'd like to get involved. Thanks.
Thanks, Kayvon. Great stuff. Next up is the epigenetics and chromatin subgroup led by Denitsa Fujimori and Alan Ashworth. And I think Denitsa will be presenting on behalf of that group.
Can you see my screen?
Yes.
Great. So it's great to have this opportunity to tell you about the ongoing work in epigenetics and chromatin and DNA replication subgroup. In our subgroup, we are interested both in host factors that are involved in chromatin regulation and DNA replication and have been identified as interacting partners of the viral proteins, as well as viral proteins themselves that are involved in DNA replication. So on the host factor side, several human proteins have been identified as interactors, including bromodomain, DNA methyltransferases, histone deacetylases, and they present potential host targets. While on the in virus itself, NSP3 protein is a macrodomain protein that represents a potential viral target. Our subgroup currently has 50 members, and we often have joint subgroup meetings with a number of other subgroups, including structural biology, drug discovery, virology, and many others. So I'm going to present two very brief vignettes about the ongoing work in the subgroup, one dealing with the host factor and the other with viral protein itself. The host factor story relates to the viral envelope protein, or the protein E, that is highly expressed in intercellular membranes of infected cells, and it interacts with a set of human proteins known as bromodomains. So these bromodomain proteins, BRD2 and BRD4, are epigenetic regulators. Normally, bromodomains interact with acetylated lysines in chromatin to regulate gene expression. And they are involved in inflammation and immune response. In that role, they are critical for antiviral defense. Thus, it was quite interesting to us to see that bromodomain proteins are also interactors of the envelope protein. What we've noticed when looking into sequence of the envelope protein is that its C N-terminal domain, one that is not embedded in a host membranes, contains what we call histone mimicry, which is a sequence that highly resembles a sequence of histone 3. There is a conservation of a critical lysine, and not far from it is an arginine that is positioned exactly the way histone 3 presents its very N-terminal motif. So our hypothesis here, and one of the models that we are analyzing, is that virus sequesters bromodomain to prevent immune response by using this histone mimic sequence to scavenge the bromodomain proteins. This is a hypothesis that is being pursued by Irene, James, Sarah, and you will have an opportunity to hear more about this work in epigenetics and chromatin breakout room. Some important implications of it are the use of bromodomain inhibitors shown here, of which there are many developed in oncology efforts to break this potential interaction. And on the viral side, a topic of interest to the subgroup is NSP3 protein, which is a novel coronavirus macrodomain protein. This protein is conserved across coronaviruses, and it antagonizes action of ADP ribosyl transferases. This protein promotes virulence and suppresses innate immune response. As such, it might represent novel therapeutic targets. In collaborative work with structural subgroup and drug discovery subgroup, of which you can learn more about in a breakout session from Stefan. Stefan in Brand's lab has looked for docking of small molecule inhibitors that bind to this domain, and he will walk you through that, as well as through a recent fragment screen with a structural subgroup that provided entry points for small molecule inhibitors. And that is all to share for this first session.
Awesome. Thanks, Denise. A great summary. Next up is the newly appointed director of the Gladstone Institute of Virology, Melanie Ott, who's leading the virology subgroup of QCRG.
Well, thank you. Hello, everybody. Welcome to UCSF, new students. Yes, I'm representing the virology subgroup of QCRG. We as good virologists are interested in the viral life cycle. A virus like SARS-CoV-2 has to enter the cell. It's a major step for the virus to gain access. It then has to hijack a lot of host factors and indirectly or directly interact with them to set up shop and reproduce its RNA, as well as produce a lot of viral proteins that then come together and form new variants to be released. So in the viral subgroup, we focusing on each of these steps and try to develop tools that can be used to interrogate each of these steps. We work with a full-length infectious virus. You will hear more about this, but we have also developed a few subgenomic non-infectious tools that can be used in regular BSL-2 environments. The first step are pseudotype viruses, which are mimicking just the entry of the virus, and you can interrogate very well what compounds or antibodies would interfere with this. We have the tools of the individual viral proteins and genetic knockdown to interrogate how this affects the viral life cycle. We can just take the viral RNA replication step and model this in cells with specific clones, and you will hear more about this in the breakout room. Or we can also mimic the late step of the virus by just combining the structural proteins and produce the so-called virus-like particles, which are basically empty shells of the virion, but can tell us a lot how, for example, host factors are important to build this late step of the viral life cycle. And we're also building up animal model capacity here at UCSF. So if you are interested in the fully infectious virus, you should visit Albert's presentation in the virology breakout room today, and he will tell you about an interesting interaction between viral protein with IL-17 and how he has interrogated this with the viral tool, virus replication studies. And yes, if you are less interested in fully infectious viruses and working in an aerosol pathogen BSL-3 and dress up in the PPE that is shown here, you might want to attend Emily's presentation, also in the virology breakout room, where she will tell you what she did to the virus and by deleting part of the structural proteins to make the virus not infectious anymore and really make it study only the RNA replication step in the so-called replicon system. But we have other systems that we are currently working on. I give you just a few examples. Dave and Joe in the Grogan lab, they are focusing on knocking down the individual factors that have been identified in the original SARS-CoV-2 interactome screen, and they're using very elaborate primary models. They're using normal bronchoepithelial cells for the knockout, and then we culturing these models at the air-liquid interface, which really makes these bronchoepithelial cells breathe and develop into fully functional epithelia. And you can see at that time, they also upregulate the ACE2 receptor and become permissive for infection. So this is a very interesting tool to study functionally what these interactors are doing. And Mali in the Frankel lab, she really focuses on the VLPs, the virus-like proteins, where you just co-express the structural proteins M, E, S, and N, and then concentrate and harvest in the supernatant the VLPs on sucrose gradients, and then you can look at them like here for other coronaviruses or here on a gel. So this is the group. We have a lot of students in the group. Beatrice, Emily, Camille, Joe, and Chris. So please talk to them to see if you find common interests. And if yes, please contact the PIs that are affiliated with the group for potential rotations. With this, I stop, and thank you for your attention.
Thanks, Melanie. Great summary of that exciting group. Next up is a more recent group, but there's been some very exciting developments in it. The cardiac group led by Bruce Conklin and Todd McDevitt, and Bruce is going to give an update of their recent work.
Okay, it's a real pleasure to represent the group today. They just start off with their this group particularly has really thrown themselves into this from the very first day of the outbreak. And we also have some terrific collaborators as shown here. I'm going to now talk just a bit about some of the progress that we've made. Really, the real question is does the SARS-CoV-2 infect cardiac cells, and which cells do they infect? And from human iPS cells, we can make endothelial cells, fibroblasts, as well as cardiac cells, which are major types of cells in the heart. And what we've found was, in fact, that we could infect them, but it was just the cardiac cells, the cardiac myocytes, which could make a productive infection. So that this is a log scale here, so that it's actually a tremendous amount more infectivity, and that we could actually see that using with a collaboration with the BSL-3 and Melanie's group. We could actually see, for instance, these are uninfected cardiac cells, these are infected cardiac cells, and you can see actually the virus, actually the double-stranded RNA just around the areas where the virus replicates. We could also see by EM the corona of the coronavirus, and which was very gratifying as well. Then we could actually then use this to test the different stages of the life cycle for drugs, which could potentially alter the effects or the infection. And what we found is actually that the inhibitors of the ACE2 did block infection as indicated here, and then also the inhibitors of the endosomal entry pathway. But interestingly, the TMPRSS2 inhibitors did not alter infectivity, indicating perhaps that a different mechanism is being used in cardiac myocytes than others to mature the ACE2 receptor. So, and this is also proof of concept that in fact that we could use this for drug screening in general. So the other thing we saw was really remarkable was that these are the sarcomeres, these are the engines that drive a contraction of the heart. But we saw that at 48 hours that we could actually see fragmentation and very, very discrete pieces of fragments that resulted during infection. And when we actually looked more carefully, we saw that they were being cut at a very specific place. So the sarcomeres are organized into these M, Z, and M lines. And we could actually see in the center of every single fragment the marker for the Z disc, indicating that it was cutting at the M lines around it. And we could even see this. This is a Z disc by electron microscopy. We could see that Z line here, the M line fragment on each end, and which was really quite remarkable, something that we have not seen before. So this creates a model, perhaps, that there's a protease which is actually cleaving essentially what holds together this machinery, and essentially this center portion, the thick filament, is actually pulled out, and that pulling out is what leaves the Z line and the thin filament. And this is something which we're trying to test now, but is really very, and consistent with that also is that there's an upregulation of many of the genes that are involved in this contact. The other phenotype that we saw was that actually that the actual nucleus was actually just severely disrupted, and in many cases gone. The DNA was gone. And we saw this in multiple cells. And interestingly, it happened in cells adjacent to the cells that are actually infected. So not necessarily in the cells that were infected, but in cells that were adjacent. Finally, we looked into, and we continue to be looking into cardiac samples, normal, healthy tissue from patients who died from non-cardiac reasons have these nice structured format, whereas patients, in fact, in this case, a very young patient, showed signs of sarcomeric disruption, consistent with the fact that there was some sort of attack. And so I'm just going to talk about just what we've shown here is that the SARS-CoV-2 will actually infect cardiac cells. And we're looking now into mechanisms of what causes this myofibrillar fragmentation. It's possible that this NSP3 viral protease that you've heard about is involved in that. And then can we observe these phenotypes in drug therapies? Can we use this for drug screens? And then where is the prevalence in this in humans as well as in primate models? So all this is actually, most of this is published in a preprint format, and then you can find out more from really the lead authors on Discord. So thank you very much.
All right, thanks, Bruce. Really exciting work from you and Todd and Juan and Mel. Next up is the protein trafficking subgroup that's being led by Shri Mukherjee.
Thank you. Can you hear me?
Yes.
Okay. So our group is interested in protein trafficking and how the virus uses the host protein trafficking pathways for its benefit. And there are two important steps of the virus life cycle that we are particularly interested. One is the entry of the virus, which Melanie and others have already talked about, its engagement with the receptor S2. And the other is how the virus then egresses out of the cell. And the reason we are particularly interested with these two steps is because if you look at the post-translational modifications that happen to these viral proteins, whether whether they're spike proteins, envelope membrane, or NSP, or nucleocapsid proteins, they all undergo post-translational modifications such as glucosylation, palmitoylation, and others, that where the enzymes are actually located inside the lumen of these secretory organelles. So the fact that they have these modifications allows us to think that maybe these proteins actually have to enter the secretory pathway for them to have these modifications, and these modifications might be very important for their functions. So when Neven published his paper with the interactome, and we looked at which proteins came down with these viral proteins, we saw that a remarkable number of these hits were proteins that localize or function in the secretory pathway. In fact, 20 out of the 26 viral proteins interact with at least one secretory pathway protein. And so this got us really excited. And what we wanted to know is the mechanism of how this virus usurps the secretory protein trafficking pathway to replicate and egress out of the cell. So the attempt we took at was to basically clone all of these viral proteins in GFP tagged vectors, both in the N-terminus as well as the C-terminus, and also as flag tagged in flag tagged vectors. And what we did was we overexpressed all of these proteins to see where they localize in the cell. And then we correlated these localization with the interacting partners that came out of that study. And we saw that while some of them did show a co-localization with the secretory organelle here shown calnexin as a marker for ER, some did not. Like, for example, these proteins didn't, but they had interacting proteins inside the lumen of the ER and the Golgi. And this led us to hypothesize that maybe some of these proteins actually need other viral proteins to enter the secretory pathway. And this has been shown for other viruses where they utilize the EMC or the ER membrane complex to recruit viral proteins inside the ER. And so currently what we are doing is basically looking to see what protein-protein interactions happen between the virus itself that allows for the entry of these viral proteins inside the cell, and what effects that has on its readout. So recently we got excited about a protein called ORF8, a GFP tag protein that had several ER protein markers that it interacted with. But this is a preliminary data from a postdoc in our lab who showed that actually the staining of ORF8 is not so much ER. It has this punctured staining, sometimes localizes with lysosomes, sometimes very perinuclear. And what it does is if you overexpress this protein in cells, you see a decrease in HLA staining, which has been now shown by another paper. They have a dramatic decrease. We don't have that much of a dramatic decrease, but it's because maybe some of these do not express as well. But this is something that we are following up. So with that, I would like to thank the three members of my lab, who's definitely the main contributors of the project, but I would also like to thank our external collaborators in our protein trafficking group, Bruno Good and Andrew Peden, who is also helping us understand the protein trafficking pathways. Thank you.
Thanks, Cherie. Great stuff. If you're interested, please join her breakout room. Next up is Eric Verdun, who's representing the mitochondrial metabolism subgroup, who's also being co-led by Natalia Jura. And I think Eric is just going to go old school and say a few words about this particular subgroup with no slides.
No, I have slides. Let me just,
you have some slides, okay.
Just share my screen. Thank you, Neven. Can you see them? Is it coming through?
Yes.
Okay, good. So I represent this mitochondrial metabolism subgroup. So one of the things that really triggered us or excited us at the beginning of this epidemic was the realization that there was a clear link with aging and fatality rate in China. And this is illustrated here on the left panel, where you can see almost exponential increase in case fertility rates as one ages. But it's not only aging, it's also the presence of underlying health conditions, even in younger people, and a clear link to risk factors as being metabolic disorders are linked to being overweight and obesity. And so this really brought into light the possibility that chronic inflammation, which we know is a risk factor for for these metabolic diseases also dramatically increases during aging might be something that would be worth looking at. And so we know that chronic inflammation increases during aging and has dramatic effect on metabolic efficiency. And so the focus of our work has been really to explore the potential interaction between SARS-CoV-2 and these two pathways. And we were immediately gratified by looking at the beautiful data set that was generated by Nevin and all of you, that there are multiple interactions between SARS-CoV-2 and metabolic enzymes. And this is illustrated here on the right side. So this led to the creation of the group and the participation from a number of people, including Akos Gerencher, who specializes on mitochondrial metabolism, and will have a small talk in the breakout room. Natalia Jura and her group, who are exploring some unique aspect of mitochondrial metabolism as well. Melanie Ott, who has studied lipid metabolism in the context of viral infection. And I want to say, so there will be a breakout talk from some of these in the next session. I want to highlight briefly another story that has emerged that generated quite a bit of excitement, and that will be discussed by Marius Walter in the breakout room as well. And it relates to the interaction between one of the key viral protein, NSP14, and protein D acylase called SIRT5, which has been known to be involved in the aging process. And so Marius will talk about this in further detail. I just want to paint a very broad picture of why we're excited by this. So SIRT5, my lab has been working on it for the last 10 years. It regulates multiple metabolic pathways via desuccinylation, demyelination, and deglycyrylation. These are novel modifications that we had identified and characterized that are present in multiple cellular proteins. SIRT5 is a major regulator. It removes these modifications in an NAD-dependent manner, and thereby regulates intermediary metabolism, but also inflammation in macrophages and other immune cells. So one of the things I would like for you to know about SP14, which is a major interactor for SIRT5. So we've been able to validate this with Marius. He will go into much more detail. We've been able to identify small molecule inhibitors of this interaction. And SP14 is a really key part of the replication machinery. It associates with the viral RNA polymerase. It is thought to be responsible for a 3', 5' exonuclease proofreading function, which is critical for this virus since it has a very long genome at about 30 kilobase, which requires a high degree of fidelity of viral replication. As I mentioned, we've validated the interaction that Nevin initially identified. And we're also working on a potential relationship between NSP14 and other proteins, GLA, IMPDH2, which are two other proteins involved in innate immunity and chronic inflammation. So I think we have found quite a rich terrain for further exploration, especially at the interface between aging and virology. Finally, one of the things that has emerged from the analysis, initial Dave Gordon and Nevin's paper, actually your paper from the whole group, is the potential identification of COVID-19 drugs that have immunomodulatory effects. And I'm thinking about mTOR as a pharmacological target. There is growing evidence that rapamycin first increases lifespan, but also leads to hyperactivation of innate immune response. And so we are exploring this pathway as well. Don't have much to report yet, but these studies are under development. And I will stop here and thank you for your attention.
All right, thank you, Eric. We have 2 speakers left. The next one is Davide Ruggiero, who is representing the translational subgroup. Davide?
Yes, thanks, Nevin. Can you see my slide?
Yes.
So hi guys, welcome everyone. So I'm speaking on behalf of the translation group. So that is a group that is involved in understanding how the virus mRNA is translated and how we can use this knowledge actually to target actually translation of the COVID-2 mRNA. And indeed, as you already heard, after the entry in the cells, a key limiting step for the virus is to translate its own mRNA or their own mRNAs. In fact, this is a positive RNA strand virus. There are key features of this mRNA. One that, the mRNA are translated through a cap-dependent mechanism, and I'll tell you in 1 second what is important. And the other one is this structure of the 5' TR of this mRNA. So this is a structure, as you can see, full of stem, loop, and hairpin. And in general, this structure is a barrier for the ribosome to scan and reach the AUG. So the virus require an RNA helicase in order to unwind this structure and allow efficient translation of their mRNA. And this will be important also in 1 second. So indeed, it's not surprising from the beautiful interactome from the Qcrg lab. So we discovered that different viral protein hijacked many translational components. Ribosomal protein, translation factor, is one that got our attention. This protein is called EF4H. Actually, EF4H is an activator of an RNA helicase. And this is what I'm talking about. So EF4H basically is part of this multi-protein complex known as 4F. And the 4H is a stimulator of 4A activity. The 4A is a dead box RNA helicase that with also EF4E and this 4G actually help the ribosome to be recruited on the 5' TR of the mRNA. And the 4A is important to unwind the structure, the 5' TR, allowing the ribosome, in fact, to reach the UG and start the translation. So our idea basically in the group is to understand how the virus actually, build the cap structure and how actually use the protein important for the cap structure actually for efficient translation, as well as how required, for example, for, on EF4A to unwind 5' TR of the virus. You will hear from Jesse from John Gross lab. They're working with Victor on NSP14, A16. These are the methyltransferase, how this is important to methylate the cap structure of the virus. And in collaboration with Shockett and the Totten lab, they're working on targeting actually these proteins as antiviral drugs. So we use different compounds that target this 4F complex that are all depicted here as antivirus drug. And the one that give a better effect, actually this drug. This is a drug known as zotatifin, or EF226. It's a first-in-class EF4A inhibitors. Actually, it's been developed by Effector, that is a company, it's the first company dedicated to develop anti-cancer drug targeting the translational machinery. And zotatifin is already in phase 1 and phase 2 as anti-cancer drug in solid patients. And another important compound that give actually very great antiviral effect is a compound known as ternatin. It doesn't inhibit initiation of translation, but elongation, because it inhibited this translation of factor EF1A. This has been developed in the Jack Totten lab at UCSF. It's also important to highlight that there is another compound, a different drug, but target also the same translation elongation factor, plitidepsin, that is also already in clinical trial for COVID-19 patients. So these are the first, this has been published, you already saw this, is the antiviral effect of ternatin and zotatifin done in antiviral assay in New York. And this is the red actually line, as you can see in nanomolar digits. Both of them, they have a very nice antiviral drugs. But you can see also that there is also some cytotoxic, cytostatic effect for this drug. And this is normal, so don't be worried. This is our cancer cell line. And in fact, as a virus, cancer hijacked this factor to build what we call the cancer proteome. So it's normal that there is some synthetic lethal effect of this drug, but the picture is completely different if you in fact actually use primary cells. This is our normal human bronchial lung cells. So you can see a beautiful actually selective index of zotatifin. It is more than 20,000 fold in killing the virus before to see any cytostatic effect the normal cells. And in fact, zotatifin now effector is at the last step with the FDA to approve, and it's almost done. And hopefully we'll start actually a clinical trial with zotatifin for COVID-19 patients next month. So if you're interested in these topics, please join our translation room. You will hear great talk. One from Jesse from the Gross Lab. We'll talk about how to target actually the 5' cap of COVID-2. Joanna Kovaski from the lab, they will tell you more how E4A helicase regulate the translation to the 5' TR of COVID-2 and how zotatifin target in the E4A. As well, I have Amar from a Gigi Lab at the Bach Institute will tell you also new features of the COVID-2 mRNA and how post-transcription methylation of the COVID-2 is important actually for its translation efficiency. So hope to see you there, and thanks.
Thanks, Davide. Really exciting stuff. I got to be in that room. I'll be in that breakout room with you, Davide.
I don't know if it's a good idea, but yeah, please join.
We'll see how it goes. Okay, so last speaker is Danielle Swaney. She's going to be representing the ubiquitin subgroup, which, as she says here in her first slide, is being led by John Gross.
Okay, thanks. Yeah, I'll try to be brief here so we can get to the breakout rooms. Yeah, we're studying, in the ubiquitin subgroup, we're studying ubiquitin, which I think is one of the most fascinating proteins in biology. And so for those of you who aren't familiar with it, it's a small 8 kilodalton protein that is can be post-translationally added to proteins. And this addition of ubiquitin to proteins involves a multi-enzymatic cascade where ubiquitin starts with an E1 protein, gets passed to a second enzyme E2 protein, and then this forms a complex along with a third protein known as E3 enzymes to find a substrate to which ubiquitin can be added to, and then process that addition to the substrate. And this can happen not just once on a substrate, but can form these multimeric ubiquitin chains. And so the addition of ubiquitin as a protein itself to a substrate can be a very powerful way of rapidly changing a substrate structure, its localization, and ultimately its function. And another layer of complexity that involves in the ubiquitin biology is these chain linkages themselves. So you can build different linkages of ubiquitin attached to a substrate. For example, the canonical linkage here, which is a lysine 48 mediated linkage. So it connects through lysine 40 at position 48 in ubiquitin itself to get this really compact ubiquitin chain structure, or you can have these elongated structure with lysine 63. And so when each of these different structures of chain structures can result in different phenotypic consequences for a protein. So canonically, K48 linked ubiquitin chains are associated with proteasomal degradation, but there are 7 lysines on ubiquitin where you can build chains from, and all of these have known different known functions. And so this is a really powerful mechanism to regulate cell biology. And so viruses are known to utilize ubiquitin signaling to promote their own replication. So one classical example is the HPV virus, where the E6 protein from HPV interacts with P53, which is the protein for TP53, and TP53 is an important tumor suppressor protein. So HPV E6 interacts with P53 to promote its degradation, and without TP53 present, you can this promotes cervical cancer, for example. And so this is one really classical example of how ubiquitin biology can viruses can utilize ubiquitin biology for their own benefit, but the ubiquitin biology is also involved in viral entry process, and additionally in the interferon response, and in many different ways and for many different viruses. And so one thing we've been looking at in the ubiquitin group is how ubiquitin is being utilized by SARS-CoV-2. And so just 2 kind of ways that we're looking at this. One is through actual infection experiments. So taking cell lines. So for example, these Vero E6 cells, which can be very easily infected by SARS-CoV-2 and either performing an infection or a mock infection and then waiting over a period of time and enriching for those proteins that have ubiquitination remnant on them and saying what are those proteins and how does that ubiquitin status change upon infection time course. And in doing these experiments, we can not only look at the host proteins and how many host proteins are regulated, what are those proteins, what sort of functions are they implicated in. We can also look at the effect of ubiquitin on the viral proteins itself, which could impact its regulation. And then we've also, from our protein interaction map, have found that some of the viral proteins interact with ubiquitin machinery as well, and looking at what is that doing? Is that promoting ubiquitin on a given substrate in its proteasomal degradation, or is this inhibiting ubiquitin on substrates, and what are those substrates that are impacted? So this is a little bit about what the ubiquitin group has been doing. And as mentioned previously, if you want to find out more about the ubiquitin group or any of the other subgroups, look at the chat box and you can find the link to Discord.
All right, thanks, Danielle. That was exactly 1 hour. That's amazing. We had, I think, 12 or 13 PIs all speak with 1 hour. I think we all deserve an award for that. So thanks, everybody, for keeping on time. So we're going to transition now to these breakout rooms in the Discord. And the idea here, just very briefly, is for especially students to jump around from room to room. And there's going to be, each group is going to be a little different, but there's going to be some repetition in terms of the presentations that are given. So they'll be repeated multiple times so that you can jump from room to room and absorb as much information as possible. And I think Gina is going to, do you have a few words of advice for everybody as we transition to Discord?
Yes. Hi, everyone. So we are entering the breakout rooms now. To get there, copy and paste the link in the chat box. Once you do that, here's an example. So once you do that, you'll copy and paste it into your browser, continue to Discord. It's loading. Over here to your left, you'll find all of the breakout rooms. Be sure to enter the voice channel, and you can enter your questions in the corresponding text channel below. And we highly recommend using the desktop version. It's much better in terms of bandwidth. So here's the desktop version, looks very much like Slack. So yeah, copy and paste the link in the chat window.
Okay, great. Well, I want to thank all the speakers again for this, and we'll see you in the breakout rooms right now. Thank you.
Okay.